Hi there, everyone. I am going to talk about the pre-lab learning on the eye and vision. So here today, I will talk about the structure of the eye and some of the vision tests which you will be performing in your McGraw-Hill Connect. So this is the diagram of the eye. Eye is located in the orbital cavity, and you can see the eyeball with the muscles, which is called extrinsic eye muscles, attaching the eyeball to the orbital cavity. So there are pairs of muscles. This is superior rectus, inferior rectus, lateral rectus, medial rectus, superior oblique, and inferior oblique. And you can see the arrows which are showing the direction eye will be moved when these muscle contracts. So these are skeletal extrinsic eye muscles. I will go over this later more in detail, but you can see there is eyeball, which is white layer, that is called a sclera. Then there is dark area here, that is called iris. Then there is hole inside it, that is called pupil. And we will talk about internal structures of the eye later. So here in the next diagram, this is the frontal eye structure. We have cut the eye from the front side and you can see the structure of the eye. So pay attention here. Eye is not a solid organ. It has wall and it has cavity inside, and cavity is filled with the fluid. So the fluid which is anterior to the lens is watery fluid and we call it aqueous humor, and fluid which is behind the lens up to the retina, that is called vitreous humor, which is jelly like fluid. So now let's go and talk about the tunics or layers of the eye. So outermost layer of the eye is sclera. Sclera is made up of fibrous connective tissue. Then the second layer is vascular. It has a lot of blood vessels and it has a lot of melanocytes which produce melanin. That's why it looks dark. And then innermost layer is retina, which is sensory neural layer. That means it is made up of nervous tissues mostly. But these three layers are not all around the eyeball. In the interior part of the eyeball, these layers are modified or absent. So sclera anteriorly is modified and becomes cornea. You see, sclera is opaque. Light cannot pass through sclera, but when it becomes cornea, that's the area which is transparent. So light can easily enter the cornea and then through the pupil, through the lens to the retina. Similarly, choroid is also not continuous anteriorly. Choroid modifies and becomes ciliary bodies. Ciliary bodies has ciliary muscles which attach suspensory ligament, and these suspensory ligament holds lens in the anterior pole of your eye. Okay, some extension of the choroid becomes the darker part of your eye, which is called iris, and there is hole in the center of the iris, that is called pupil. This is the only area where light rays enters into the eyes. Okay, So these are the modified structure of the sclera and choroid. The yellow layer here, which is the retina, is not even reaching the anterior part. So mostly the posterior and middle part of the eyeball inner wall is covered by retina, and we will see them in detail in next slides. This is the eye model, which anterior half of the eyeball is removed and you can see posterior part. So as usual, you have sclera here, then the choroid, and this pinkish layer is here, the retina. If you see in the posterior wall of the eye from inner side, you can see certain spatial area. You can see there are a lot of blood vessels. These blood vessels supply the nutrition and oxygen to the retina. Then there is a light area here, which is the optic nerve starting point. And in this area, there are no photoreceptor light receptors. That's why we call them blind spot. Then there is a small large dark area and a small white area in the center. So this large circular area is called macula lutea. And the center small white area is called fovea centralis. Macula lutea contains mostly cones receptor, and fovea centralis contains only cone receptors. Cone receptors are for the daylight vision or for the color vision. So once any image from outside falls on the retina, you can see the object, but to see them very clearly, that image must fall on the macula lutea and fovea centralis. Here is anterior of the eye from the inside. So earlier we saw posterior of the eye from inside, and now it is anterior of the eye from inside. So you see there is hole that is called pupil, and the connective tissue which is covering it is cornea. Around the cornea you have iris, then the lens, and then ligaments for the genial fibers and ciliary muscles. Here the same structure we talked earlier about, and now you can see there is three layers: sclera, choroid, and retina. And then this is fovea centralis, optic disc, optic nerve start from the optic disc, and in the center of the optic nerve you see blood vessels, artery and veins are entering and supplying the retina. This is again the models. You can see the sclera and the extrinsic eye muscles, then iris, and then pupil, and cornea will be here. Layers of the eye we talked earlier, three layers. This is the eye dissection. This is the cow eye with the anterior pole of the eye and then muscle and fibrous tissue and fats attached to the eye. And then once eye is dissected, you can see the structure of the eye. So this is interior half of the eye, this is posterior half of the eye. If you see from inside of the interior half of the eye, you can see the lens. We have removed the lens, so lens is here. Then you have iris, this dark area, and this hole is pupil, and in front of the pupil there will be cornea. And then if you see posterior wall, you can see sclera, choroid, and inner gooey like a structure, fatty structure is the retina. Okay, don't worry about dissection, we do that in a human anatomy. This is the way showing how you dissect the structure and function of the eye. You can see here. So let's go and talk about each layer. While both the sclera and cornea are making up the tough outer lining of the eye and serve to protect it, both sclera and cornea, only the transparent cornea allows light to enter the eyeball. So this is only cornea which allows the light to get inside. The iris or choroid does not. The choroid is the second layer and supplies sclera and retina and all associated structure with oxygen nutrients. So the main function of the choroid is to provide oxygen and nutrition. Retina is the innermost layer of the eye and contains photoreceptors, and two photoreceptors are rods and cones, and other nerve cells necessary to detect and process the incoming light. It is flattened against the eyeball by the vitreous humor. So vitreous humor lying here flatten the retina, otherwise retina will detach easily and becomes squeezed or crumbled. The one area in the back of the eye without any photoreceptor is optic disc. Here is no photoreceptor. In all other area you will have photoreceptor. This optic disc is the starting point of all the nerve fibers, axons coming from here and become the optic nerve. So it is the exit for all optic nerve fibers, and we call it also the blind spot because there is no receptor. Axons from ganglion cells in the retina become the optic nerve which transmit all visual information to the brain in your occipital lobe. Same structure here. The iris, I talked earlier about these structures. Okay, rods and cones. So this you see here, this is the retina, and in the retina you can see rods and cones. So this is inner part of the retina, means towards the cavity part of the retina. You can see cones. C for C, cones are responsible for color vision, daylight vision. Rods are for dim light or dark vision. Okay, so two kinds of photoreceptor cells are rods and cones. Cones detect color light, and there are three kinds of cone: blue, green, and red for each type of colors. The retina's photoreceptor cells are the rods and the cones. The more numerous rods can be stimulated in dim light and react to light sensitivity. So what happens in the uh, night or during the dim light? A lot of rods get stimulated once and then absorb whatever light is available, and that you can see the object but not so clearly like daylight. This is again the parts of the eye after dissection we talked about. Layers of the eye, we have already talked about it. Lacrimal structures. One of the function of the eye is to produce tear and clean and bathe your eyes, because eyes are exposed to atmospheric air which can be very cold, very dry, or exposed to several dirts and microorganisms. That's why it needs to be cleaned every minute. So lacrimal gland lies on the lateral superior part of your eyeball, under your eyelid. They are exocrine gland. They secrete tear and then drain through the small ducts and then pass into your eye, and it bathe your sclera, cornea, and all your eyeball, and then drain through the medial canthus through superior and inferior canaliculi. After draining from there, it drains into lacrimal sac, which carry the tear into nasolacrimal duct and then ultimately pour into your nasopharynx, and then it goes into your stomach. Most of you have experience when you put eye drops in your eye, it is drained ultimately into your throat and you can taste them sometimes. When you are emotional, your gland controlled by autonomic nervous system and you cannot control your tear secretion. In that time you produce a lot of tear. Your superior inferior canaliculi cannot drain them all. And that's why the tear drops come outside when you are crying. Okay, so that is the lacrimal structure. Some of the kid, the newborn baby, are born with blockage of this nasolacrimal duct, and in that case tear comes all the time. Those babies are like tearing all the time. In that case you need to clear this nasolacrimal duct to drain the tear. They do by surgery. Okay, so some of the test of vision, the visual acuity test. uh, in this test you are asked to stand 20 feet away from the Snellen chart, and Snellen chart contains the image or the letters, and then other side you have several number. So a person with normal ability to focus far away object will clearly see all symbols on a Snellen chart at least up to row 8. You can read up to here. This is called 20/20 vision and means that the lens focus distance object right onto the retina. So when you are standing 20 feet away and when you are reading this, the image, you see light image, light rays focus the reflection of these letters pass through your cornea and then focus on the retina. Then you can see clearly. If it goes behind the retina or in front of retina, then you cannot see them. It will be blurry. So let's see if you can see this line 8, which is 20/20. That means most of the normal individual can see that line from 20 feet, and you can also see that line from 20 feet. That's why your vision is normal. Okay, let's see, you are standing here on the 20 feet and you can see even a smaller line, which is line 9, and that is 20/15. So if your vision is 20/15 means you can stand at 20 feet and you can see this line. That means you can see that line number 9 from 20 feet, but most of the normal individual needs to comes to 15 feet, which is nearer to the object to see that object. That means your vision is excellent, better than other normal individual. So the Snell chart as seen by someone with myopia, which is called short-sightedness. In that case, image focus in front of retina. Instead of retina, it will focus here in front of retina. If someone suffer from myopia, which is nearsightedness, they can see the symbols well, and depending on how far in front of the retina the image is focused, they will lose the ability to recognize the letters. So let's see, if you are in 20 feet, you cannot see these lines. You can see this one only. Okay? Because image is focused on in front. So what do? Then you do put a glass, and then glass will adjust your bending the light and ultimately focus on the retina so you can see it. Objects close to the eye appear blurred, image focus behind the retina. So a person with farsightedness or hyperopia, this is hyperopia. What happens in this case? If you are suffering from hyperopia, instead of focusing on the retina, it will go behind the retina. So a person with farsightedness or hyperopia will do okay with Snell test. But if they have to look at something close up, when you have to read the newspaper, you will have trouble reading it without corrective lens because your image will fall behind. And that's why you need the converging lens. So I talked about understanding the visual acuity result 1. So let's see somebody's vision is 20 by 20. This line you can read from 20 feet. So each line on a visual acuity chart has a pair of numbers at the left margin. These numbers are on top of each other. The upper number is always 20. This refers to the fact that we stand 20 feet away from the chart. So this is, you stand from 20 feet, all 20, 20, 20. And then which line you can see. Let's see if you can see 20 by 20, that means your vision is normal. If you can see only 20 by 50, this line from 20 feet, what is the meaning of that? Guess it. You have 1 minute. You can think about it. Stop the video. Think about it. That means you can see this line from 20 feet, but most of the normal individual will see from all the back here from 50 feet. So your vision is not good. So understanding visual acuity result, smallest print a person is able to read from 20 feet. So this is 20 by 40 and this is 20 by 10. We have explained it and you got it. Another visual test is test for astigmatism. So astigmatism is caused by the uneven shape or surface of the cornea and the lens. So when your curvature of the cornea is irregular or uneven, then you will see blurry object. Some part of the object you will see clear and some part blurry. So the world will have strange curves and blurriness for a person with severe astigmatism. A lot of kids born with it. So to test that, we use Astigmatism chart. And when you see them, you can see here, one of the line, if you have defect, you will see clear and other lines will be blurry. So the horizontal line is 0 degree and from there, which line is clear, you can measure. So this is 120 degree. Okay. So determine the angle of lines. 0 degree is horizontal, 90 degrees vertical. And then doctor will put different kind of lens in front of your eyes and find out how much astigmatism you have, and based on that they will put the lens, and the lens required for correction of astigmatism is irregular or uneven lenses. So normal appearance of the astigmatism chart, which is also called Green's chart, you can see, you will see all the spokes of the wheel, but seen with astigmatism, what do you see? This line number 1 is clear, but other spokes are not clear. near accommodation and the lens. So you see, focus on distance object. So you are focusing on the distance object and see the size of the lens here in number 1 and number 2. So accommodation is the quality of our eyes, and that is performed by function of the ciliary muscles and the lens. So accommodation is the changing of the shape of the lens that occur when normal eye focused for close vision or far vision. In order to focus something close by, the lens must become rounder and bend the light more. So you see here, the light is coming and you are focusing on the far object, which focus on the retina, and the diameter of the lens is less. But now the muscle is relaxed and lens is thicker. So near object converged through the lens and focus on the retina. Okay. So for the lens to rebound elastically, the muscles of the ciliary body must contract, and that loosens the suspensory ligament, allowing the lens to bulge or become thicker. And that's why you focus on the, as you old, what happened? These muscles, ciliary muscles, cannot function the same way, and you cannot read, and you need glasses. Near accommodation and the lens, near point accommodation chart. So younger you are, your ciliary muscles and everything is very efficient. So with your average near point in centimeter is good. Like you can see, if you're 10 years, you can see 7 centimeter object. If you're 20, 10, 30, 13, 40, 20. You see after 40 how it changed? That's why most of the people need glass after 40. 50, 45, 60, 90. That's why you have seen your grandpa or grandma, when they read book, they put far away. Even they're not wearing sometimes glasses. Some people are good eye, but still they need to put their uh, book far away from their eyes to see them clearly. This is the color blindness uh, chart, which is called Ishihara chart, and you can find the color blindness. We are not doing this chart. This is the chart, Snell chart without glass, and this is with correction of the glass, and then how you see the same chart. This is the Astigmatism test. We explained about it. This is the change in near point accommodation, and again the color blindness. So you can see normal vision, you can see here 12, 8, 16, 36. Try to read those numbers and try to find whether your eyes can see color completely or not. Okay, so this is the blind spot. We talked about it. Pupil. Pupil controls the amount of light to get inside your eye. So if this is very bright light, then our pupil constrict and they allow only limited amount of light. If it is dim or you are looking at the far object, there your pupil is wide open and more light get inside it, so you can see them. Okay, so these are the in the eye. Now we have two eyes, so when you see one object, both eyes see the same object. To make two the view one, our our eyes move towards the midline. So axis of the eyeball moves toward the midline slowly and converge both image in one, and that is called convergence reflex of the eye, and so that you can see one object clearly. Okay, that is all. Thank you very much, guys.